Okay, let's talk about what factors can determine the rate of sedimentation of a particle during your centrifugation. Okay, sedimentation is the main thing that applies to the particles you have in the sample, right? So what are the main factors that determines how fast things will be settled down as a pellet? Okay, number one is of course the centrifugal force. Okay, centrifugal force. The higher the force is, sedimentation will take place much faster. Number two, the viscosity of the medium. Okay, so viscosity means how dense your solution is, the medium is that's holding your sample. Okay, there should be a difference in the viscosity between your target sample and the medium, the solvent. Okay, if there's no difference, you'll never get any pellet coming down and never get anything sedimented down as a pellet. Okay, so if there's a big difference in the viscosity between your target molecule and the solvent, yeah, the sedimentation will take place much faster. That's what it means. And the size of the particle also matters. The larger the particle is, there'll be more higher rate of sedimentation that's going to take place. And then difference in density, like I just told you, viscosity, density, okay, between the medium and the sample. The difference is very important. The bigger the difference is, the sample will come down as a pellet much faster. Okay, so S is the sedimentation value. Okay, and then when you use your centrifuge, the rotor is the important part because that's where your sample will be located, and that rotor will be the main thing that will create the centrifugal force because it'll be spinning around. Okay, so the rotor, okay, has a driving shaft. Okay, and the rotor itself, and the motor of course will spin down this entire rotor, of course. Okay, and depends on the speed of your centrifuge. Okay, you can call them low speed centrifuge, high speed centrifuge, or ultra speed centrifuge. Okay, low speed is up to normally about 10,000 RPM, creating about 8,000 G force. High speed is about up to 30,000 RPM, which creates about 100,000 G force. Ultra centrifuge can go up to 120,000 RPM. That is about 700,000 G. Okay, so there are various types of centrifuges out there. Ultra speed centrifugation normally involves creating a vacuum. So you spin at a vacuum field because it creates a lot of resistance. So it has the vacuum pump attached to it. And the most commonly used centrifuge you are familiar with will be the microcentrifuge that you use in the lab. Okay, only spins about between 1 and 2 milliliter sample. So where do you use your centrifugation? Okay, so number one is preparatory centrifugation. What does that mean? You are trying to prepare some biological molecule by going through multiple centrifugation. That's what it means, preparatory centrifugation. So think about it. So you have maybe tissue cells. Okay, if you maybe grind them up, maybe if you break them up using physical force, like homogenizer, okay, you can break up their cell structures, okay, and then you spin at the low speed, about 10 minutes to 800 G. Then you can separate out maybe the pellet and the supernatant. In the pellet, you will have heavy, large, chunky stuff like nuclei and cell debris and stuff. And you can transfer the supernatant to a next centrifugation tube and spin at a higher speed, maybe 20,000 G, about 15 minutes. Then you can also gain pellet versus supernatant. In this time, pellet will include much of organelles like mitochondria or chloroplast, okay, and the rest of them will be floating in the supernatant. Again, transfer that to a new centrifugation tube, and you spin again at the higher speed, 100,000 G, about 1 hour. Then again you can get a pellet. This time the pellet will be made up with smaller things like microsomes, okay, and pieces of plasma membrane and internal membranes. Okay, again you transfer the supernatant and then spin at the ultra centrifuge. okay, like 150,000 G, 3 hours. Then what can you get at the end? Ribosomes can be separated. So if you are studying ribosome, what they are made out of, ribosomal protein and ribosomal, RNA, you can separate those out. So these continuous separation we consider as a preparatory centrifugation. Depends on the speed of your centrifugation and then centrifugal force. You can separate out different things from your cells at different time point. Okay, makes sense? And the analytical centrifugation is you can also determine the molecular weight, purity, shape, and other physical characteristics of molecules using your centrifugation. Okay, it depends on the molecular weight and the size, their sedimentation. S value may be different, so you can measure the S value. So that's what analytical centrifugation is. If you learned about molecular biology, there's a classical Okazaki experiment where Okazaki fragments during the DNA replication process is identified. So in that experiment, they used analytical centrifugation. They identified the smaller replicated strand, and which is smaller than the longer replication strand. Okay, so yeah, if you have a chance to look over that experiment design. And then modes of centrifugal operation. Number one, differential centrifugation. So like the previous case, there's a big difference between the target molecule and the supernatant in the density, so sedimentation value, right? So you will have a pellet and supernatant easy separation. That's what differential centrifugation is. Okay, so sediments tend to have unsedimented contaminants, and the yields of the smaller particles are low. So smaller particles will be floating on the top as a supernatant, and the large molecules will be settled at the bottom. Okay, so pellet versus supernatant, that's what differential centrifugation is. The initial material will be separated out between the pellet and the supernatant. Okay, and the density centrifugation, it is a way of separating out your molecules based on their density. So when you do that, you need to create a gradient of density in your centrifugation bottle. Okay, so by adding heavy dense materials like sucrose or ficol, you can create a layer of sucrose or ficol solution, maybe 20%, 10%, 5%, 2%. Okay, you create different like layers of densities, and then your sample will be added somewhere, and then you centrifuge. Then your sample will end up at the place where it has the same density as one of those layers. Okay, so that's what density centrifugation is. Okay, it depends on the density difference. You will separate out your molecule. So that's the example. And the density centrifugation method can be divided into rate zonal centrifugation and BIOT density centrifugation. Okay, so rate zonal centrifugation is, like I said, you create a different density, layers of different density, maybe 17 grams per mil up to maybe 11 grams per mil. Okay, and then when you have a mixture of sample, if you centrifuge, your sample will be separated based on their density. Okay, in the separation method, particles are separated based on their sizes and masses rather than density. Okay, so since you create different different zones, they'll end up at different zones based on their size and masses. Okay, rather than the density. That is the centrifugation condition you provide will make a difference there. And another way of doing that is BIOT density or isopynic centrifugation. So it is based on the density, not size. So you create a uniform density and you separate it out using something like cesium chloride solution, and you can get them separated based on their density. So again, the difference is, the previous one, rate zonal centrifugation, you start out with a different rate, different zones in the beginning, and your molecules will end up at the same, the zones, they are same as their density. But here, you don't have those different layers at the beginning, but the layers are formed at the end through the centrifugation. I guess that's the main difference in between those two methods. Okay. And then there's a method called continuous centrifugation. So when you have to centrifuge a large sample, let's say you have 100 liter to centrifuge out, or 1000 liter from your bioreactor, there's no way you can use a micro centrifuge for that volume, right? So you need a better design to handle a large volume centrifugation. So this is the design that can handle a large volume sample. So the design will allow you continuous sample loading. Okay, so your sample will come in while the centrifugation is taking place. So there's a rotor being spinning, and your sample will be introduced here. Okay, and number 5, your sample is coming in, the mixture of whatever the things. And then as soon as they are applied to centrifugal force, what's going to happen is the separation will take place. Heavy large stuff will end up on that place where the pellet will be sitting in. Okay, but since there's a gap there, they'll be going through the hole and they'll be collected here, the large heavy stuff. Okay, continuous collection of that pellet will take place, and the light things that will be floating as supernatant will go over there, and the excessive volume will be captured through here. So number 2 is the tubing where the supernatant will come out continuously, right? So there's a continuous feeding of your material, and continuous collection of your pellet and continuous collection of your supernatant is possible. So through the continuous operation of centrifuge, you can even separate out about 100 liter volume if you use this method. Okay, it's the design of the operation that allows the large-scale centrifugation. And then instrumentation within your centrifuge. Of course, the rotor is the most important part where the sample will be loaded, right? And rotors will come in different shape. They may come in as a horizontal rotor or swinging bucket type. So sample can be hanging here, and when it spins, it kind of swings around like that 90-degree angle. So that's a horizontal rotor or swinging bucket kind of design. Okay, it swings and it kind of moves like that. And then another design is called fixed angle. So there's no change on your sample position. So it'll be just placed right there on the fixed angle. So your pellet will end up there. And compared to this, your pellet will end where? Right at the very bottom, right? But here, the heavy pellet will be loaded right there, because that's the place where the maximum centrifugal force will be applied, because centrifugal force is relevant to the size of your radius. That's the largest radius size. That's where the largest centrifugal force is applied. So that's where the pellet will end up. Okay, fixed angle loader. And then this is a vertical loader. So it's a vertical design. The sample will be added. So the same radius here, top and bottom. So your pellet will end up on the side of the wall. Okay. And then when you use a centrifuge, the operation-wise, the most important part you need to be careful is balancing the rotor. When you add your samples to the rotor, okay, you cannot have just one sample added, because it should be counterbalanced here and there. You will remember how you use the microcentrifuge. You have to have even number samples that are equal weight. So you create a balance on the weight on your rotor. Otherwise, you may see something like that. This used to be a centrifuge, but since somebody loaded the sample without balancing it, during the ultra centrifuge operation, the entire rotor was damaged on the very high centrifuge condition. So this could creates an environment where people can seriously hurt. Okay, so you definitely do not want to create this kind of error. Okay, make sure you balance. Counterbalancing is very important. Okay, before your centrifugation operation. And then rotor is expensive as well. It's normally made out of expensive metal like a titanium. So you need to take a good care of your rotor, so prevent any corrosion. And you have to use the right speed for your centrifugation. You wash it well, rinse well, dry well, 70% alcohol wash in water, disinfect them well. And derator speed when spinning very dense sample. Derating means you remove braking. Okay, that's what it means. So sudden stopping will hinder your centrifugation. Your pellet may be disturbed. Okay, read your box 3222 for more information about care and what cares you need to provide your centrifugation. Okay.